And we were the first group in the world to show that stem cells in ovarian cancer are definitely what cause the metastasis, and they're probably the ones that get transformed, and they are the causative agent of ovarian cancer.
Our lab is totally the diverse. Basically, what we are trying to address, that as I said, they are deregulated proliferation. We are asking why.
My lab specifically focuses on the role of oxidative stress in cancer progression.
My lab and majority of the clinicians also, they're expecting to identify the new biomarkers, which can not only help for the diagnosis, it also can guide the clinicians for the better therapeutic approaches.
Now, why does it occur? And what one knows is that each of the differentiated cell in our body actually has a very definitive lifespan wherein it performs specific functions. For example, everyone knows, even school children know, that the lifespan of a red blood cell is roughly 120 days. So after that, it is going to reduce its efficiency of transporting oxygen, and then it will soon undergo a process called as apoptosis, which is programmed cell death. So it means that there is a time span in which it will live, it will function, and then it will die away. So then how are these cells again replenished? You have stem cells that are very rare cells present in the tissues of each organ, and these then are activated by some of the micromolecular signals that stimulate them and trigger them into cell division. They will then give rise to that critical mass of cells, progenitors as they are called, that will give rise to the differentiated cells again, and which will go again into the tissue at their specific locations and perform the functions that they are destined to do. So this is a nice cycle of events that occurs in all tissues in the body, in all organs in the body, and their normal functioning is what is referred to as normal tissue homeostasis. So that is why we move, we function in the way that we do. Now sometimes, these cells revolt. Now this type of a revolt can happen at any step. It could be the stem cells themselves, that may be the recipients of, say, some insulting events in the environment, which gives rise to genetic alterations. Again, you can also have some inherited defects which could give rise to perturbation in this homeostatic mechanism. But what happens is that some of these cells, they may either just, apart from proliferating rapidly, they fail to differentiate. So it could be the stem cells that proliferate rapidly or the progenitors that rapidly proliferate. And then what happens is, because there are a fewer number of differentiated cells, the functioning of the organ is also suboptimal. So there are fewer differentiated cells to actually perform the functions of the organ. So this is actually the crux of what causes cancer.
It is the unlimited proliferation of the cells, and then this will form that one mass, okay, and then with time they will be aggressive, and then at the source, the origin where the tumor just initiated, with time it will be malignant, it will be nearby lymph node, it will be spread, that is the malignancy, and then it will come to the circulation, and depending the source of or origin of the cancer, it will go to the specified organ that the secondary tumor forms, and that is the metastasis. And once it is reached, no return. Whatever the present scenario is, that what is the diagnosis, no more. Depending on the cancer, 1 week to 2 months. So this is a unique disease in that sense.
The cancer is a disease, okay, so that involves abnormal cell growth, and it also has the potential to invade into the other parts of the body. And the major the cancer arises from, a normal cell, it will become the malignant cell. It is a multi-stage process, and it is a kind of a transformation of the normal cell to the malignant cell.
You could have a set of symptoms that can be captured in the clinic, but to understand them and to treat them effectively, you need to actually look at what is happening at the molecular, the cellular level to come up with better therapies. And I think the past several decades are witness to the fact that cancer research has achieved a lot of has achieved a much deeper understanding of the disease, the cells, and the different types of molecules that they exhibit, which can be harnessed to target treatments that will be effective. So I think cancer research thereby becomes very important.
Cancer is the second leading cause of death globally after heart diseases. It still is the same condition. The molecular mechanisms are incompletely understood, although globally research is going on continuously to explore newer targets, newer ways of treating the cancers. But still, lot needs to be done. So, considering that, we need to identify pathways or molecules which can be specifically targeted. Why specifically? Because considering the chemotherapeutic drugs that are in use, there is a lot of, as you may know, that there are a lot of toxicity to the normal cells. So we need to understand molecular mechanisms, and by understanding that, we will be able to identify targets which only can be targeted in case of cancers, so that we avoid the normal cells, or the normal cells can be spared.
See, we are the basic scientists. We understand how it is happening, and what are the main. And then we want to discover the molecule and drug, and those are coming to the clinic. And clinic, the doctors, based on those markers, what we basic scientists discover, based on that, they decide that. But I think if we amalgam that scientists as well as the clinicians, there may be more fruitful outcome will come, much more. See, they will know the pathology more, we know that cause more. So if we know both together, sit together and understand this, then it will be much better and it will be much faster. What we do, because we don't have access, we don't do that, the patient's things. But when we also observe those things, get that our that knowledge will be also there. This will be more appropriate also to understand the pathology side. When we do the basic scientist, we basically do that why it is happening, cause. But when it is happening, how the phenotype, how the patient feels. Suppose we discover a drug, how that the compound we have discovered, that may be good, but how the patient's response, those we are not getting. There is a huge gap between scientists and clinicians. So I think that bridge, in the US what is happening, many of them, of the scientists, though they are doing the basic research, but they have training with that, the medical MD, okay, so they know the pathology also. So that's the important, the basic research, as well as the clinicians, amalgamation, both should be in the same table and discuss to root out, means find out the causes, and then accordingly we can discuss. So that's why the cancer research, it will continue, because as I said, it is a unique disease. In a same family, if 5-6 people are the patient of same cancer, I am not talking about different types of cancer, that is again ball game. Okay, same cancer, 5 people may have the different region. So that is so complex. Okay. So it takes time. It takes, see, the people say that it is cancer has no answer. That is not true. That is not true. If we detect in the benign stage, almost there is a chance of 90% recover. 90% recover. And for that, we need to know each and every type of cancer, a panel, the very critical. And that maybe in India we have diversity, we can test those and then find out that commonality. Okay. So that will help, research will help. More clinician, more scientists should come together.
So if you want to go for the screening every year after certain age, after 45 crossing the, for example, ladies, so there is a chance of breast cancer. But the problem is coming is the whatever the testing, the methods what they use, it is very expensive. Okay, like in the CT scan, mammography, these are very expensive methods. And so the research is also going, can we diagnose the cancer, or at least can we screen the cancer by using a non-invasive methods, by using the less expensive method. So research is also going for the cancer research, that one.
At NCCS, we have a very good history of doing, or we've been one of the earliest groups in the country to do molecular research in cancer, right from the inception of the institute over 30 years ago. I joined around 20 years ago, and at that time there was no group in India working on ovarian cancer. Most of the focus was on breast cancer, which is indeed which affects a large fraction of our population. Cervical cancer is another thing that is quite India-centric. Ovarian cancer occurs at a much lower frequency. But there's a lot of research that goes on in breast cancer, cervical cancer, and there has been a huge amount of information that has been gathered regarding not just their molecular behavioral patterns, but how these can be tackled better. Ovarian cancer within India was a kind of an orphan disease, I think. I would like, just for the to give it a name, it was a kind of an orphan disease. There were no groups who were really working on it. So I thought that why don't I take this up as a challenge, especially because it's often called as a silent killer. It has very few symptoms, and by the time the patient is diagnosed, the disease has advanced often to stage 3. And at that time there is metastasis, which brings the patient to the clinic, and that is almost incurable. So the mortality with ovarian cancer is probably one of the highest in all of the gynecological cancers, and it is comparable only with the triple negative breast cancers, which is a subtype of breast cancers which are very aggressive. And there is the similarity between ovarian cancer and triple negative breast cancers. So that is why I started to work with this. I thought that was something that needed to be looked at even in the Indian context. And I started off by making resources in the form of cell lines, animal models to study the disease, and so on and so forth. And in doing so, we actually identified, because we were working with a lot of primary tumors from patients, and we found that there seemed to be some cells within these tumors that were virtually indestructible. They look beautiful under the microscope, unfortunately so. And it was fascinating to actually look at these cells, and I could sometimes imagine them just moving around in the patient's body and causing havoc. But to look at them under the microscope was sheer pleasure. We started thinking that probably these have a stem-like phenotype to them, and that is how we actually were the first to report we did a lot of studies. Of course, we had to develop a lot of assay systems and to really show that these have a stemness-like features within them. And we were the first group in the world to show that stem cells in ovarian cancer are definitely what cause the metastasis, and they're probably the ones that get transformed, and they are the causative agent of ovarian cancer. So that was an achievement we made way back in 2004, In fact, it was the third fourth tumor type worldwide to be associated with stem cells, or what are now known as cancer stem cells or stem-like cells in cancer. We have a lot of terms that are now evolved for these things. But that was a very fascinating thing, and I still remember the first sample wherein I saw those cells, and I thought that, I don't know whether we can really think of these as stem cells, because they have the regenerative function, which you remember I told you, the normal stem cells in the body, they keep on regenerating with the purpose of repair and replenishing the differentiated cells, whereas these cancer stem cells, they seem to be replenishing the tumor cells so that they actually do not die. So thereby the tumor becomes indestructible. Isn't that a sorry state of comparison? But indeed it is the truth. Then we've done a lot of studies on those aspects. Thereafter, we even actually would tag these cells in animal models, of course, with a fluorophore, which was a vital dye, that is, it would not kill the cell that was stained, and then with a simple principle that this stain would get diluted out between two cells after every division, and at a certain point of time it would just undergo quenching, so it would not be detectable. Now, you'll remember I said stem cells actually stay dormant in a tissue, and they are activated only when there is a need for cells to be replenished. Similarly, in tumors, these cancer stem cells also stay quiescent or dormant, and they actually evade most of the chemotherapeutic drugs because they are quiescent. Most chemotherapeutic drugs target rapidly multiplying cells, so that is why they evade it. And then, of course, because of the inherent regenerative capability, they can give rise to a tumor. So how does one detect dormant cells is always a challenge. So we came up with this, wherein we were labeling it with this fluorophore, and then within tumors we would identify using a process called as flow cytometry or flow based sorting. We would sort out these cells which maintained the fluorophore, yet when we sort them out, bring them in culture, they could give rise to tumors. So this was a proof of principle, and it was really very fascinating. It was very satisfying, let me say, to actually do all these things, wherein we put forth a theory and we can show that it does exist. Now the next challenge in the field actually of cancer stem cells is how do you target them? Because they evade most of the commonly used chemotherapeutic drugs. So how does one actually do this? What we did is that we isolated these, took the membranes, because in terms of a therapeutic drug, we feel that it will be more effective if it targets any molecule that sits on the membrane of the cell rather than an internal target. So we took the membranes and we raised monoclonal antibodies against the proteins that are present in the membranes of these cancer stem cells. We screened a lot of hybridoma colonies that we had developed, and we came up with actually 3 or 4 candidates, but one of them actually is a monoclonal antibody that can target very specifically the stem cells in a tumor as well as the progenitor cells. So this is something that we have developed in the last 10 years, and it's now, we patented that, and we hope that it will be very useful in the future. So I think this definitely shows how basic research is required if we need to target cancer in the clinic. So this is just one of the examples. Now when we were doing some of these studies, we also moved on to the omics type of profiling, wherein we've been looking at the protein profiles, that starting off with the genome, the transcriptome, epigenome, as well as the proteomic profiles. And then we realized that one of the problems in cancer treatment is the fact that a tumor is not a homogeneous group of cells. Although they may arise from a single cell, by sheer way of the rate of proliferation, the fact that some cells are going to stay quiescent, Some of them even differentiate and give it a characteristic histopathology that the tumor is associated with. And so it's a kind of a diverse group of cells that are present within that structure, which is often talked of as an abnormal organ sometimes. So because of this, even the molecular patterns that are expressed are not homogeneously expressed. So even though we have a monoclonal antibody that will target a certain protein, it may not be present on all the cells in the tumor. So by just evasion, of expression of that particular target molecule, some of the tumor cells may again remain behind. So we came up with an approach. In fact, it was Rutika Naik, a very bright student in the lab. Using flow cytometry, we actually came up with an approach wherein we could stratify or bring down the tumor or deconstruct the tumor into groups of cells, each group of which will have a different level of regenerative potential. So thereby we could actually use this to predict under which group of cells are being targeted by a certain chemotherapeutic drug, and then by use this scheme again to achieve a combination of different drugs that can achieve remission. Remission is complete treatment of the tumor wherein it is retracted and it will disappear. As a proof of principle, we've done this using animal models, wherein understanding this intra-tumor heterogeneity really helped us a lot. So this again is another example wherein we try to understand why we are not efficiently able to target tumors even in the patient, and what can be done. What are the different approaches? Increasingly when we look at cancer cells, we appreciate the cunningness with which they survive in the body. Every time you have an approach, there are always some cells which escape it. And that is why, sometimes the treatments do fail. And there is a never-ending type of mechanisms that can come into play for the type of treatment that is given. So a cancer cell, it's a very challenging adversity. So they will adapt themselves to the type of microenvironment which will be toxic to their survival, and they will come up with mechanisms wherein they will survive and then again come back into play once the environment is changed. That is when treatment is stopped. So it's kind of a ticking it's like a kind of a ticking bomb that sits inside the body and waits to rear its head and give rise to the disease again. So it's a challenge all the time to keep on, it's a play between two adversaries. So it's very interesting, and that's what actually keeps us alive. All our thoughts and our approaches, we come up with newer things. So right now we have come up with something which is not even published right now, but we have been working on it for the last 3 or 4 years, something called as chimeric transcripts. Now each one knows the central dogma, which is you have a gene, it gets translated into RNA, which is the transcript level, and then that may get translated into a protein, or the transcript will remain as a non-coding RNA which will perform very specific functions. Now sometimes what happens is that you can have a transcript that emerges from 2 genes. And then these chimeric transcripts give rise to proteins that are completely novel. So this is what we are studying. Why? Because these are likely to be very tumor specific. One of the disadvantages of some conventional treatments is that while targeting the tumor cells, they also target the normal cells of the individual, and thereby we hear of so many different types of side effects. Now, if we could find out some very tumor specific molecules in the cell, then that could be an approach to give a very targeted treatment which will be effective only in the cells that express those molecules and not in the rest of the body. So that is something that we are developing right now, and we found out some of these molecules we've also found that they are actually immunogenic. A few of them, we have about 55 molecules that are likely to be immunogenic. Now the next level is to test them out, see to what extent they are expressed because of tumor heterogeneity, and let's see how we can go ahead with this. So this is something that is coming up. Hopefully in the next 5 years we will have some leads from these type of projects. So it's research that keeps us active actually, and I hope I have convinced you that there's a need to keep on doing research to better understand this disease.
After, for example, if the age crosses 50 or 60, it is highly in the like European and US countries, they go for the screening. But we don't have that system, and it's also very expensive. So one of them, even the my lab and some of the labs is working, can we identify screening biomarkers? So that means if the patient knows, okay, before the symptoms comes, okay, so then he can go for again further the advanced techniques to confirm the whether cancer, and also the treatment they can immediately start. Okay, so the treatment requires the very less, more effective treatment and very less expensive, that one. And there is a very less chance of the relapse, there is a very less chance of the mortality. So that is the way, again, the research is going to, again, identify a non-invasive screening biomarkers and also the diagnostic markers. And the third important point is also the majority of the cancers you see, it is relapsing. Even the treatment is general 3 types of the treatment they do. One is the surgery, second is chemotherapy, and the radiotherapy. Even after these treatments, a majority of the cancers are relapsing. So it is also, the clinicians also requires if they know there are various alternative strategies treatments are available. But they use for the everybody once diagnosed, they give the first to go for the a common treatment strategy. So, but the clinicians already knows, okay, this treatment may not work for this particular patient. So they can give the alternative treatment. They can save the patients. But for that also, there is a required for the therapeutic biomarkers, which can guide the clinicians during the treatment, a kind of a prognostic markers. Okay, so the research is also going, even including the my lab, to identify a therapeutic markers, which can guide the clinicians for the better treatment strategies, and that may be the step towards a personalized medicine. Okay, where we can know that this patient, this kind of treatment strategy can work, and this strategy may not work. It's not a difficult task. Actually, the cancer already started to develop the kids. Okay, but the problem is, so again, so the cancer is a very complex disease, that. Okay, so how the research is going on, one of the approach, what we understand is we use a multiomics approach. Okay, so we do with we collect the clinical samples and we look for the what proteins are altered. Okay, in case of the breast cancer, where we have the collaboration with the local hospitals, we collect the clinical samples, we look for the what proteins are altered. And similarly, we also look for the what genes are altered, what metabolites are altered. So if we identify some correct protein marker, which is altered only in the particular, the breast cancer, that one. So we can develop the kit that we can use for the screening as well as the diagnosis, that one. But the major problem is coming on majority of the biomarkers so far identified, it has a less sensitivity and also the less specificity. So it is not only marker for the one cancer. Okay, so it is also marker for some of the other cancers. So that is what a particular cancer, to develop a particular kit, is becoming the challenge. But the research is going on, now every year we are coming with the FDA is approving some of the biomarkers which can be used for the particular cancer. But the strategy is the same. How they are identifying the kits they are making is, first we have to go with what proteins are altered in particular cancer. Okay, and then we can, it's a kind of any biomarker discovery, we have the 3 steps. The first step will be through discovery. So where we take with the clinical samples and try to identify what proteins are altered in that one. Then we go for the validation phase, where we take the large cohort of samples and we target only that particular protein. We check it out using the method like ELISA, Western blot. Okay, several methods we use to target, to look for that particular protein in a large cohort of sample. If we are success, then we go to the next level, clinical trials, where we can, if we are succeeding in the clinical trial, then we can able to develop the kits for that one. But majority of where we are failing is the discovery phase. We are coming with so many markers, and also validation also we are coming up that one. But when we are going to the clinical trials, the specificity and the sensitivity is really lacking in the biomarkers. But it's not like that the research is not going. So in the NCCS, National Cancer Institute, So they have started to look for the so many biomarkers. Okay, and we are also, some of them are really successful to come up with the biomarkers using the multiomics approaches. One of the cancer we are working in my lab is the multiple myeloma. So multiple myeloma is a disease is caused with the plasma cells. Okay, the plasma cells will generate the abnormal antibodies, unfunctional antibodies. So we have the collaboration with the Armed Forces Medical College. So what we are trying to identify biomarkers for the multiple myeloma. And again, the key thing in the multiple myeloma is like other cancers. So when the patient come to the clinic, and once it is diagnosed, that is almost in the later stages, and the treatment is not effective, and the maximum survival rate is the 5 years. Okay, and there is a multiple myeloma having a 3 stages. Like one is the benign form, one is asymptomatic form, third one is the symptomatic form. So what we are trying to identify is, can we identify a biomarkers which we can diagnose at the asymptomatic stage or the benign form. Okay, so we are trying to identify the we collected the clinical sample for the 3 type of the patients, and we are trying to we do it with the proteome profiling. So in the first stage, what we did is we compare the proteome profiling with multiple myeloma patient versus the healthy control. Okay, we have done with the extensive quantitative proteome profiling, and we identify the panel of proteins. At least 4 protein panel we are identified. So including whatever the one of the proteins, albumin, is already known as a marker, that one. So we now currently we are trying to develop a biomarker. That panel we are validating in the large cohort of samples with the benign as well as the asymptomatic form. And another, we also, not only the biomarker, we also look for the new targets. Okay, so what we have done is, see, this multiple myeloma is occurring in the bone marrow. So we collected the bone marrow patients, sample from the multiple myeloma as well as the controls, non-hematological malignancies, and we look for the what proteins are altered in the bone marrow intestinal fluid of the multiple myeloma patients. So we identified some of the very interesting protein. So one of the protein we identified the MZB1. So it is a marginal zone B cell specific protein. And again, multiple myeloma causes from the B cells only, because B cells produce the plasma cells, mononuclear cells produce the plasma cells. And this MZB1, even if you look in the functions of the MZB1, so it involves in the immunoglobulins, and it plays a major role in the calcium homeostasis. And multiple myeloma, the patients is mostly the abnormal immunoglobulins will be generated, and the patients, once you come to the clinic, when the bones are fractured, that is only the symptom, that one. So bones are fractured means that the calcium homeostasis is affecting that one. So we have taken this MZB1 protein and we validate in a large cohort of sample. So now we are also identified that we compare with the non-multiple myeloma cell line versus normal cell lines, B cell lines. And now when we knock down that MZB1, so what is happening again? It is the cell proliferation is decreasing, apoptosis is increasing. That clearly confirms that MZB1 is a potential target for the multiple myeloma, which we identified in our lab. So now what we are trying to use, we are trying to understand mechanistically what MZB1 playing the role in the multiple myeloma patients. So that also research, the even continuous research is going to understand the mechanistic aspects of this biomarker before going for the advanced stages. Okay, next level. So we are trying to look for the mechanistic aspects of the MZB1.
Our lab is totally the diverse. Basically, what we are trying to address, that as I said, they are deregulated proliferation. We are asking why. Why? See, there is, see, the cell division is happening in the 4 phases. Okay, each phase has the checkpoint. If something wrong happens this phase, the next phase should not, your cell should not go to the next phase. So the checkpoints are there. Okay. Our study is that how this, and in the cancer, these checkpoints are deregulated. Okay. We are trying to understand how it is deregulated. Okay. What are the main component which are involved in the deregulation process. Okay. And so identify those, find out that what is their status in the normal versus cancer of different grade. If any key molecule or key genes we identify, then design the drug molecule against them. So what our study is revealing that there are many more components which basically activates this kind of oncogenes. And what oncogenes generally normal cells, if there is some defect cells will die, and there's a cell death mechanism are there, the apoptosis, programmed cell death, autophagy, all these things are there. And these cancer cells, they are very clever, they block those, the death pathways, block those so that these cells proliferate much more, make the tumor. And mystery in this, in our cancer research is that when it is malignant, it is going 4-5 tissues, organs, and this small mass is forming, and how this small mass actually interfering the normal functions of that organ, that we don't know how it is happening. And we believe that this is one part, is that cell cycle defect. We are interested to know that how cell cycle defects happening. And another thing is that how signaling pathway, that many of the cancer, their signaling pathway is totally deregulated, and they actually helps to breach this checkpoint. Okay, like for example, the lung cancer, the EGFR pathway is the MAP kinase pathway is very important. Colon cancer, that RAS signaling pathway is very important. Okay, so they are deregulated. Okay, we are trying to understand how they are getting deregulated in cancer. Because this kinase, if you aberrantly you block them, then what is happening? You can prevent the metastasis. Okay, then you can help that clinicians, that okay, this is the things if it is there. And most of the cases drugs are there, but drugs are so cytotoxic, the patients actually face huge unbearable pain. So in future what will happen, we have to think about the formulation and drug which will have minimal effect. Because what is happening so far, the drugs, they are also targeting that the toxicity effect is coming, because of that normal cells also getting affected. So I think in future people are trying that it should not affect the normal, or minimal and maximum target specific effect is there. Okay, so we are trying to that how cell cycle is deregulated, how these kinase pathways are also getting deregulated, and mostly focus on the protein post-translation level. Why? Because each cells, the workforce is the protein. Okay, protein. So protein, if it is, and as I said, the cancer, less than 10% that mutation is responsible. Mutations happening after means that after development of cancer, most of the mutation is coming. But during initiation, very few, and there is no specific marker is there, this will be, that is less than 10% cases is there. We believe, our group believe, whatever happening, it is the happening at the most of the cases is the protein level. So if we know the protein levels functions, and if we find that how protein is getting deregulated, then we can restore their function, so that we can stop the malignancy. So that's why our lab totally focus on the protein level, which is the functional unit of the cell, each of the cells, how they are getting deregulated and why they are getting deregulated, who is deregulating. Okay, so that's the main focus of my lab, and we are trying again the understanding that how that this protein, when it is a malignant phase, how they are actually cross-talking the organ and impairing the organ normal function. So that is our major concerns. See, if you stop that, okay, this would not impair the functions of the organ, then organ will function, then the patients will survive. So that is our major concerns actually.
So reactive oxygen species are known to play a very significant role in almost all the hallmarks of cancer. My passion and my work has always been focused on the role of oxidative stress in diseases, and of course I have been very curious to know what is happening in cancer. Different cells have different types of free radicals, or they can generate different types of free radicals. Their sources can be different. There are different various sources of free radicals. Then there are depending on the concentration, free radicals can be either a signaling molecule which is required for our normal functioning, or if they are generated in a higher quantity, they can be damaging to the system, causing diseases. So that fine balance has to be maintained in the cells. Somewhere this balance is dysregulated in case of cancer or any kind of diseases. So one of the important sources of free radicals in our body are, of course, mitochondria. Is one of the major sources. We need to look into primary sources of reactive oxygen species, and there are enzymatic sources. Of course, there are enzymatic sources also. So, for example, NADPH oxidase, which we commonly call NOx, and then there are lipoxygenases, cyclooxygenases, monoamine oxidases, etc. There are many enzymatic sources. Again, except NADPH, all others are secondary sources. Okay. NADPH oxidase is the only primary source in our body which is required for generation of free radicals for the signaling mechanisms. Anything that, any normal body function, we need signaling, and they act as signaling molecules. So that's the only primary source of reactive oxygen species. So cell lines, there are less aggressive and more aggressive cell lines. And I was curious, what happens, why do some cancer cell lines are less aggressive while the others are more aggressive? And these are the more aggressive ones are the cause for the triple negative. These are the triple negative breast cancer, which is difficult to treat. And aggressiveness is the cause for this. So I wanted to understand what is the role of oxidative stress in these cell lines, in the tumor aggressiveness. So I started addressing, and of course, as I said that NOx is a primary source, I wanted to see how NOx is playing a role in this. So if you want, I can explain you a little more about what we have observed and how it is interesting. So we started with a normal breast cancer cell line, breast cancer cell line which is less aggressive and a more aggressive cell line. And when we saw the expression of this enzyme, it was amazing. So what we found was, as literature showed, that NOX4 is overexpressed in many cancer cells. We found that the less aggressive cancer cell line has more expression of NOX4, and the more aggressive has less expression. Okay, it's like normal cell, but still there is a difference. So that was very interesting. By understanding the types of free radicals, we thought that by understanding the types of free radicals, where they are located, what splice variants they have, and what interacting partners they may be, that will shed more light on why there is an aggressive behavior. So in vitro and vivo, so we use in vivo system also, and then patient samples also. By comparing all this, we will be able to assure that yes, some molecules do play a role, and then probably now with the tools like bioinformatics and other things, we will be able to find more inhibitors for these. If once we identify, then it's not easy that you start with randomly you start using inhibitors on this thing. If you can find more inhibitors and then validate, that will be really a great So as I said, reactive oxygen species, role of reactive oxygen species is one area, one outlook on this, and that I am focusing on.